In 2018, the world watched in awe and apprehension as news broke of the first gene-edited babies. Chinese scientist He Jiankui announced the birth of twin girls Lulu and Nana, whose genomes had been altered using CRISPR technology. He Jiankui aimed to confer HIV resistance upon the twins by disabling the CCR5 gene. This unprecedented event marked a turning point in human history, igniting a firestorm of ethical debate and scientific scrutiny. The birth of Lulu and Nana thrust the once theoretical concept of designer babies into the harsh spotlight of reality. It raised profound questions about the limits of scientific ambition, the sanctity of the human germline, and our responsibilities to future generations. The scientific community largely condemned He Jiankui's actions, citing insufficient safety protocols and a lack of ethical oversight. The incident served as a stark reminder that the power to manipulate the very building blocks of life comes with immense responsibility. It also underscored the urgent need for comprehensive ethical frameworks and international collaboration to govern the use of gene-editing technologies. The genie was out of the bottle. The world had to decide how to manage its incredible power. To understand the controversy surrounding designer babies, we must first grasp the groundbreaking technology that makes it possible: CRISPR-Cas9. CRISPR, short for clustered regularly interspaced short palindromic repeats, is a revolutionary gene-editing tool discovered in bacteria. It acts like a pair of molecular scissors, allowing scientists to precisely cut and paste DNA sequences. CRISPR-Cas9 has rapidly become a cornerstone of genetic research, offering unprecedented speed, affordability, and accuracy in manipulating genetic code. This revolutionary tool empowers scientists to target specific genes within an organism's DNA, either disabling them, correcting mutations, or inserting new genetic material. Its applications span a wide range of fields, from agriculture and biofuels to medicine and biotechnology. In medicine, CRISPR holds immense promise for treating genetic diseases, developing novel therapies, and understanding the complexities of human biology. Its ability to precisely target and modify genes has opened up a world of possibilities for curing inherited diseases and even enhancing human capabilities. However, this newfound power also raises profound ethical questions about the potential for unintended consequences and the blurring of ethical boundaries. The potential of CRISPR technology extends far beyond the realm of theoretical debate. It is already making tangible differences in the fight against human disease. Researchers are actively exploring its applications in treating a wide range of genetic disorders, from cystic fibrosis and sickle cell anemia to Huntington's disease and muscular dystrophy. One remarkable example of CRISPR's therapeutic potential is its use in treating beta thalassemia, a debilitating blood disorder. In a groundbreaking clinical trial, researchers successfully used CRISPR to edit the faulty gene responsible for beta thalassemia in patients' bone marrow cells. The modified cells, once reintroduced into the patient's bodies, began producing healthy red blood cells, offering hope for a life free from painful blood transfusions. CRISPR is also being investigated as a tool to combat cancer by enhancing the effectiveness of immunotherapy. Scientists are developing CRISPR-based approaches to modify a patient's immune cells, making them more adept at recognizing and destroying cancer cells. These advancements signal a paradigm shift in medicine, offering the potential for personalized, gene-based therapies tailored to each patient's unique genetic makeup. While the therapeutic applications of gene editing inspire hope, the prospect of using CRISPR to create designer babies has ignited widespread controversy. The term designer babies refers to the hypothetical practice of using gene editing to select for desired traits in offspring, such as intelligence, athleticism, or physical appearance. Proponents of designer babies argue that it empowers parents to give their children the best possible start in life potentially eliminating genetic diseases and enhancing desirable traits. They envision a future where genetic engineering could eradicate inherited diseases and even enhance human potential. However, this line of thinking raises profound ethical concerns, prompting questions about the very definition of normal and the potential for exacerbating existing social inequalities. Critics argue that designer babies represent a slippery slope towards eugenics, a dangerous ideology that seeks to improve the human race through selective breeding. They fear that allowing parents to choose their children's genetic traits could lead to a dystopian future characterized by genetic discrimination and a widening gap between the genetically enhanced and the unenhanced. The ethical implications of designer babies are vast and complex, demanding careful consideration from multiple perspectives. One of the most pressing concerns is the potential for exacerbating existing social inequalities. Access to expensive gene editing technologies could create a world where only the wealthy can afford to design their children, potentially creating a genetic underclass. Furthermore, the very notion of selecting for desirable traits raises questions about what is considered normal and who gets to decide. What one society deems desirable, another may find objectionable. The potential for cultural bias and discrimination based on genetic makeup is a serious concern. Another ethical dilemma revolves around the issue of consent. Future generations will have no say in the genetic alterations made to their embryos, effectively denying them the right to an unedited genetic inheritance. This raises profound questions about parental autonomy versus the rights of the unborn child. Finally, there's the significant risk of unintended consequences. Altering the human germline, the genetic information passed down through generations could have unforeseen and potentially irreversible effects on future populations. Our understanding of the human genome is still incomplete, and even seemingly minor genetic modifications could have cascading effects on other genes and bodily functions. The ethical and societal implications of designer babies have sparked a global debate, with different countries adopting varying stances on the use of gene editing technologies. Some nations, like the United States, have implemented strict regulations prohibiting the use of gene editing for reproductive purposes. These regulations aim to prevent the creation of designer babies while allowing research into the therapeutic applications of gene editing to continue. Other countries, such as the United Kingdom, have adopted a more permissive approach, allowing for limited use of gene editing in embryos for research purposes. The UK's Human Fertilisation and Embryology Authority, or HFEA, has granted licenses for gene editing research on human embryos to study early human development and improve in vitro fertilization techniques. However, the lack of a unified global consensus on gene editing raises concerns about reproductive tourism. Individuals seeking to circumvent stricter regulations in their home countries may travel to countries with more permissive laws to access gene editing services. This highlights the need for international cooperation and the establishment of ethical guidelines to ensure responsible development and application of gene editing technologies. The rapid pace of advancements in genetic technology, particularly CRISPR, makes it crucial to engage in thoughtful and informed discussions about its potential implications. As we stand on the cusp of a new era in human history, we must proceed with caution and a deep understanding of the ethical complexities at hand. One of the most pressing challenges is to establish clear and enforceable ethical guidelines for the use of gene editing. These guidelines must strike a balance between fostering scientific progress and safeguarding the sanctity of human life and the well-being of future generations. International collaboration is crucial to ensure that these guidelines are universally adopted and enforced. Public engagement and education are also essential to ensure that decisions about gene editing are made with input from all stakeholders. Open and transparent dialogue about the potential benefits and risks of these technologies will be crucial in shaping public policy and ensuring responsible innovation. The potential implications of designer babies extend far beyond the realm of individual families, touching upon the very fabric of society and the future of our species. The ability to select for specific genetic traits could have profound consequences for human diversity, social equality, and the very definition of what it means to be human. Imagine a world where parents can choose their children's physical characteristics, cognitive abilities, and even personality traits. While this may seem like science fiction, it could become a reality in the not-so-distant future. Such a scenario raises profound questions about the potential for genetic discrimination, the commodification of human life, and the erosion of human dignity. The widespread use of gene editing could also have unintended consequences for human evolution. By artificially selecting for certain traits, we risk narrowing the gene pool and making future generations more susceptible to diseases or environmental changes. The long-term impact of such interventions on human health and evolution remains largely unknown. The dawn of the gene editing era presents humanity with both unprecedented opportunities and profound ethical challenges. As we navigate this uncharted territory, we must proceed with caution, humility, and a deep respect for the sanctity of human life. The decisions we make today will have far-reaching consequences for future generations, shaping the very essence of what it means to be human. We must engage in thoughtful and inclusive dialogue about the ethical implications of gene editing, ensuring that all voices are heard and all perspectives considered. This conversation must extend beyond the walls of academia and research labs, engaging the public in a meaningful way. The future of gene editing is not predetermined. It is a future that we will shape through our choices and actions. It is a future that demands our collective wisdom, compassion, and an unwavering commitment to the well-being of all humanity. Let us proceed with caution, guided by the principles of ethical responsibility, social justice, and a deep reverence for the miracle of life itself.